course, when we look at the respiratory system, we know that there is the main function of the respiratory system is for gas exchange, getting oxygen into the lungs, which will then move it into the blood and be carried to the cells to make energy under oxidative phosphorylation. And so we need a continuous supply of oxygen. In that process, we make carbon dioxide, and we need to transport that from the cells through the blood to the lungs and out. So that is the major function of the respiratory system. Another couple of functions that will be somewhat applicable to our interest is there is olfaction, smell. And since that is in the nose, part of the respiratory system, we will look at what is called the olfactory epithelium as part of our discussion. And then the vocal cords for vocalizing are in the larynx, which is part of the respiratory system. So we'll kind of deal with that a bit as well. When we breathe in and breathe out, of course, we're breathing air in through the nose and also the mouth into the pharynx, which is often subdivided and then it comes down to the larynx trachea. We have a little epiglottal flap that when we swallow, it covers the glottis, which is the opening of the larynx trachea, so that liquid and food do not go down the larynx trachea to the lungs, but instead go posteriorly to the esophagus. So we'll look at the epiglottis as well, and then we'll look at the larynx and trachea, which is really a single tube, but the initial part we term the larynx, and then the continuation is the trachea. And we're going to see that we have a number of areas of cartilage in there. The trachea eventually bifurcates into two structures, and this is the bronchus. The bronchi is plural, the bronchus, and then that enters the lungs, and then the bronchi branch into the bronchioles, and they get smaller and smaller airways. We'll see that the bronchus has cartilage in the wall, and the bronchioles do not. These airways go to smaller and smaller structures. Finally, we get to the so-called terminal bronchioles, which go to the alveolar ducts. The respiratory bronchioles, the alveolar ducts, the alveolar sacs, and the alveoli. So we'll be looking at all of those structures as well. Remember, we also have a pleura around the lungs, right on the visceral layer of the pleura. This shows you a mid-sagittal view. In the nose, we have these little structures called conchi, right here, or turbinates. There's 3 of them, and we'll see some view of those. And we have mostly respiratory epithelium, which is pseudostratified ciliated columnar epithelium. But there's a patch in the upper part, the superior part, that is called olfactory mucosa, which looks very similar to the respiratory mucosa, except it doesn't have goblet cells and it has olfactory neurons for smell, which will then go to the cranial nerve 1 olfactory nerve. So we'll look at some of those structures. Then you can see that the nasopharynx is designating pseudostratified ciliated columnar epithelium that we see here. That's also, we can just say a little bit more briefly, we can call that respiratory epithelium. Then in the oropharynx, it changes to stratified squamous, non-keratinized, and then that sort of continues there. And then as we get down into the trachea, the upper part of the epiglottis is pseudostratified squamous non-keratinized epithelium. The underneath part is respiratory epithelium. Then we go through the larynx, and we go through some false and true vocal cords. The false cord is also called the ventricular fold. You see that there, and the true cord is the vocal fold. And so we'll see that we do some transition from stratified squamous to respiratory and back there. Then we get into the trachea and its respiratory all the way down until it gets a lower and lower height to the epithelium. We have a number of different cartilages. We have elastic cartilage in the epiglottis, and in the larynx and trachea we have hyaline cartilage. That's what this is indicating. We have a epithelium that goes flatter and flatter as we get way down deep into the lungs. So we're seeing that here. These represent the goblet cells, and where they kind of finish up. And this is your goblet cells, how far they go. And the cilia and all of these things, it shows you the parts of these structures that have these structures within them. This is showing it more in a table form, and I know when you see a table it's a little overwhelming, but a lot of this is for reference, so if you want to check any kind of the features here. Notice, for instance, we don't have any goblet cells in our olfactory mucosa, but we do in the respiratory mucosa. So there's just a little indication of information we could find by looking at this table. This is our very common respiratory epithelium. Since we feel mellow today, we'll mellow out and look at his stuff. This is pseudostratified ciliated columnar. And of course we can see the cilia very clearly here. We've looked at this epithelium many times, and here we see it. Here we see some goblet cells. There. These are supporting serous and mucus glands underneath. And so we're quite familiar with this epithelium. So we've seen a lot of this in the respiratory system. What are these? These are the nasal conchae, or the turbinates. So if we go back, that's these structures right here. You see that one, that one, that one. Anyway, those are the conchae, and here they are. Inside the nose, this is what they look like. We have this little structure. This is a weird stain. I shouldn't say weird. It's not an H&E stain. This is the epithelium. You see the cilia here, the epithelium. What is this? This is a little bit of bone in those conchae, or turbinates, is the other name. And so that's what that is. Here's more bone off to the side of the nose over here. So this is within the nasal passage, and this is what they look like. Now the olfactory epithelium, where is it? I think it's helpful to go back and look. This is right here. This is our olfactory mucosa. Well, what is the mucosa? What is the epithelium and all these other places? It's respiratory, right? Pseudostratified ciliated columnar. But this patch right here is a little different, and in the lab we should be able to differentiate that. This is the olfactory mucosa. So when we get to the olfactory mucosa, what do we have? Well, somewhat in the center of that epithelium, we have the olfactory receptor cells. They're going to have some projections up to the surface. They're chemoreceptors, and they will transduce smell into action potentials in cranial nerve 1 or the olfactory nerve. Okay, that's coming here. So these are the receptor cells. And then the nuclei of the supporting or sustentacular cells. You can see these in blue. They're kind of columnar shaped, and they really span the entire olfactory epithelium, but their nuclei are up higher than the other cells, so they look like they're more towards the apex, a little higher. So these are supporting cells, and then at the base we have the aptly named basal cells. The basal cells can undergo differentiation and turn into these olfactory receptor cells if the receptor cell gets damaged or wears out. So now this is what it looks like in reality. This is now an H&E stain, and our olfactory epithelium, as you see from our bracket, goes from here to here. And we will generally have both cilia and microvilli on there, but we won't be seeing goblet cells. This shows some ducts of the so-called Bowman's glands. These are olfactory glands, the olfactory or Bowman's glands. They're kind of mucus, but they're a little bit of a watery mucus, so they kind of stain a little darker than most mucus glands. So they stain kind of darker here. But that's the stuff that goes in the nose and gives this mucus, sort of watery mucus secretion there. Now, we're at the bottom here. We would have the basal cells, and then these are the olfactory receptor cells, and then at the top here, the nuclei are the supporting cells. We'll see some very often nerves that are part of the olfactory nerve, cranial nerve 1. And you see that in locations here. There's a little arteriole there. And we'll often see some venous sinusoids. There's another arteriole right there. We can see, and so we see those structures that we're fairly familiar with. This is another view. So our olfactory epithelium goes from about here up to here. Here we're seeing the cilia. The microvilli we don't really see well under a light microscope. These are the nuclei right here, and this layer that we're kind of tracing across, that's the nuclei of the supporting cells, sustentacular. These nuclei here in the mid-range are our olfactory receptor cells. So that's these. And then down here at the base, we have our basal cells that are coming there. Then we can see a lot of venous sinuses here, view out. Lots of blood there. You can see some red blood cells in that one. And we see some nerve from the olfactory nerve. And then we see, oh, here's some more nerve. And then we see some of these glands, these kind of watery mucus, kind of look a little cirrus-like here, and those can go out to the surface. So we'll look at that a bit. This shows it again with the diagram. Sustentacular or supporting cells with their nuclei at the upper layer, and then the nuclei in the middle are mostly the olfactory receptor cells, and then basal cells at the bottom. Here they are again. These are your Bowman's glands, these mucus, but kind of look more serous, and they're going to secrete to the surface. Here's your basal cells, the nuclei. Here's your olfactory receptor cells, and then this upper layer, the nuclei of the supporting cells. These are your venous sinuses. You can see the red blood cells in the lumen. There, we caught one without any red blood cells, but you see there's one more in longitudinal here and a little bit here. So we're seeing that. There's a little bit of nerve here, but we're not seeing a lot of nerve in this particular image. This is a little higher magnification. Again, the sustentacular cell nuclei have this columnar shape, and then here's the nuclei of the olfactory receptor cells. They have two processes, so they are bipolar cells, so we can also call them bipolar cells. We saw bipolar cells in the ganglia, the spiral ganglia in the inner ear. We'll also see them in the retina eventually, and we have them here. These are some of the places where we have the configuration of bipolar cells with the processes coming off each end, like here and here, for instance. There's a venous sinusoid there. And of course, here's your basal cells down here. This is an electron micrograph, and so we can see both the cilia, and here's the shorter microvilli. Cilia and microvilli. We're not going to ask you from an electron micrograph to differentiate between the sensory neurons and the sustentacular cells, but there, it shows you them labeled there. All right, here's the epiglottis. That's that flap of tissue that slides over the opening, the glottis, leading into the entrance of the larynx trachea. We've looked a bit at the epiglottis when we looked at elastic cartilage, because in its interior, which we see right here, it's labeled EC, this is elastic, a cartilage, so we'll see that. And then on the upper surface where there's more wear and tear, we have non-keratinized stratified squamous, and then on the underneath surface, we have respiratory epithelium. So we have a transition there. As we go down into the larynx, we see the vocal cords. We're going from proximal, closer to the nose, if you will, to distal, closer to the lungs. So it's coming down in this direction. And as we're going down through the lumen, we first come to these false vocal cords, also called ventricular folds. So we have respiratory epithelium that changes right about there to stratified squamous, non-keratinized, and then it goes back to respiratory, and then it comes back over the true vocal cord to stratified squamous, and then transitions back to respiratory. So where you have the wear and tear on the vocal folds, you're going to get stratified squamous. Other places it's respiratory. Now it would be nice if when we're looking under the microscope, it would be always that way, but there's a lot of exceptions to that, and there's sort of transitioning back and forth. So we'll do the best we can to see it, but that shows the transition. So these on both sides are the ventricular folds. Then we go into this little space here. We call that a sinus, and then we come back over the true vocal cord or the vocal fold. So ventricular fold is the false, and then we have the true vocal cord, which is the vocal fold, and that's this. Sometimes when we're looking at it under the microscope, some of this will be artifactually removed during the processing, so we won't always see it come to a nice point like that, because of course you could see how that small piece could be easily sort of lost in processing, but sometimes we'll see. In this true vocal cord, this vocal fold, we have a vocalis muscle, and so you can see the muscle on both sides. We also have some glands here, and so we'll be looking at that as well. We have skeletal muscle in those areas, and of course it's the vibration of this true vocal cord, vocal fold, that gives the sound, changes the frequency, and gives a different pitch to the sounds we were speaking and singing and that kind of thing.
Where's the transition zone between the non-keratinizing stratified squamous and the respiratory?
So it goes respiratory, and then as you're approaching the fold right about here, it switches to stratified squamous, and it comes over the fold like that, and then as you're getting into the sinus, it switches back to respiratory around here, and then right about there, it switches back to stratified squamous as you go over the fold. And right about somewhere in here, it's gonna go back to respiratory. So it goes back and forth. So if you think of it as stratified squamous over the folds, respiratory everywhere else, that's pretty close to telling us where it is. And if we remember that stratified squamous is better for wear and tear, and the folds are going to have more wear and tear on them, that might help us keep that straight and remember it. This shows you another view. This is the false vocal fold here, the ventricular fold. There's the sinus, and then here's the true vocal cord with the vocalis muscle on both sides. This is cartilage that you're seeing over here to support it. And so that's kind of what it looks like. And again, you may not always see all this intact when you look at it under the microscope. It's a little hard to keep it all without messing it up when you make the slide. But hopefully we'll see enough of it. This is showing the stratified squamous over the vocal fold. And again, you say, well, these are flat, but some of them are kind of rounded. And you can see a little bit of variation there. But we would call this stratified squamous right here.
Is that considered parakeratinized?
No, this isn't even considered keratinized. So there's no keratin here, so it's okay. We have the nuclei coming to the surface, but there's no keratin.
Okay.
So this is all non-keratinized. Now, having said that, sometimes with irritation, it can keratinize. It can transition, right? And sometimes our respiratory can transition to stratified squamous if you get a lot of wear and tear. And so we call that metaplasia, don't we? Where you get some wear and tear. So if we see in reality, there may be patches of stratified squamous or even keratinized where we would expect to find respiratory, for instance. And that could be just the animal or the individual that they took the slide from. We can see some variation. We'll see quite a bit of variation when we get to the urinary system going through the urethra, for instance. Sometimes you can get 3 or 4 different types of epithelium going down through the urethra. So there is places where there's quite a bit of variation. And we do see some of it here in the respiratory. But, because you might be tempted to say, oh, that looks more like transitional because look at how round these are. Well, we wouldn't really call that transitional. We'd still call that stratified squamous, but it's kind of in between stratified squamous and respiratory. It's kind of a little bit of a in-betweener right here. Now here's a very abrupt transition. This is respiratory in the sinus. See the cilia here, and look right about here. It goes to stratified squamous over the fold, and this nuclei get flattened. So sometimes that transition is really abrupt. It kind of reminds us of the junctions in the digestive tract, the sphincters, where it could be very abrupt. But sometimes it's not going to be that nice of an abrupt transition. It'll be a little bit more gradual. As we breathe in down the airways, we have what are called the conducting zones. That conducts the air, but we don't get any gas exchange, so we don't get any benefit of that air. If you remember your physiology, that makes up the dead space, that air. We won't get too much into that, but that just to give you a little throwback. Starting here with your respiratory bronchioles, your alveolar ducts, your alveolar sacs, your alveoli, all of these are in what are called the respiratory zone. The respiratory zone is named that because that is your starting gases. You're putting oxygen from the lumen of these structures into the blood, and you're taking carbon dioxide from the blood and putting it into these areas. So this again shows you your conducting zone, and over here, starting with your respiratory bronchioles, your alveolar ducts, your alveolar sacs, your alveoli, that's all the respiratory zone, and you have gas exchange. Now, the alveolar sacs are clumps of alveoli. It looks like a cluster of grapes. If each alveolus was a grape, that would be an alveolus, and then the whole cluster would be an alveolar sac. We have about 8 million alveolar sacs in the body, in each lung, and we have about 300 million alveoli. Remember, alveolus is singular, alveoli is plural, so that's what we're talking about there. So from here on, we have gas exchange, so we call this the respiratory zone. The prior, the area proximal to that, is called the conducting zone. This shows an image of the trachea, and of course, right adjacent to it is the esophagus. So this is the esophagus over here, and this is the trachea. We see the cartilage, this is hyaline cartilage, and so we see that there in the trachea. Here it is in black and white with the same image. Now, this is, don't be confused, this is thyroid gland. On Friday, when you look at that audio lecture on the endocrine, you'll look quite a bit at the thyroid gland. It just has nothing to do with the respiratory system. It's just close to the trachea and the esophagus in the neck. And so here it is. This is thyroid and this is thyroid. There's even a little bit of parathyroid down here. And so over here is a little bit of parathyroid. But this is the trachea. That's what we're mostly interested in now. The trachea has rings that we can feel, and these are made out of hyaline cartilage, and those are these cartilaginous rings. We call them C rings because they make the shape somewhat of a C. So in this case, it looks like kind of a backward C. The opening between the ends that make the space that make the C is at the back of the trachea. So this would be the posterior part. And in that opening of the C-shaped hyaline cartilage ring, we have a little smooth muscle called trachealis muscle. And that can help with a little bit of contraction of the trachea. So there is a little bit of trachealis smooth muscle right there. Now this is low magnification, so we can't tell, but this is respiratory epithelium coming around here, and then here's our cartilage and around. There's our esophagus back there. So here's our trachealis muscle, smooth muscle patches, and it often looks kind of patchy like that. Here's the epithelium again of the trachea, and again it's our respiratory epithelium, and we have some blood vessels. We have both mucus and serous glands in that area that can secrete into the lumen, and here's our hyaline cartilage that makes up the C ring cartilage, the perichondrium on the outside, and then the cartilage. With the lacunae and the chondrocytes, and on the edge here we have the chondroblast. This is a nice H&E image of the respiratory epithelium of the cilia, and here we see some nice, these happen to be serous glands. We can see both serous and mucus. Here's some venous sinuses. See the endothelium here that we feel more familiar with now, the endothelial cells flattened. And here's our cilia and our respiratory epithelium with the basement membrane there. As we go into the trachea branches into two bronchi, a right bronchus and a left bronchus, and it enters into the lung as a bronchus, a right and left bronchus. And the bronchi, we can tell them from the bronchioles in the lung because the bronchi have still cartilage in their wall. They also have smooth muscle, and they have respiratory epithelium, but they have cartilage in the wall. The bronchioles, which make up the bulk of the airways in the lungs, are missing. They no longer have cartilage. They have a more prominent layer of smooth muscle and of course they have respiratory epithelium as well. This shows you a bronchus over here. There's the cartilage in the wall. We're in the lung now. This happens to be an artery, pulmonary artery here. You can see the blood inside of it even, and the endothelium there. This is the respiratory epithelium. This is a different stain. This is periodic acid shift with iron hematoxylin. So a little bit different staining there. This is smooth muscle right here. And then here's our cartilage. Here it is with the black and white imagery. Now, as we're inside of the lung, intrapulmonary bronchus, again, we will have the respiratory epithelium, we'll have cilia and all of these, but we'll have some endocrine producing cells as well, a number of different cells. This is our first good look at the lung tissue. When we look at the respiratory system, we'll look at the nasal passage, we'll look at the trachea, the larynx and the trachea, and then we'll look at the lung slide, and it'll look kind of like this. This shows a bronchus. How do we know? It's got hyaline cartilage in the wall. It also has smooth muscle. We're sort of tracing it around there. That's smooth muscle. And of course, the smooth muscle can help constrict and dilate the bronchi and the bronchioles. These are bronchioles. No more hyaline cartilage. No more cartilage in the wall. And so these are bronchioles. These are actually down here, terminal bronchioles, which are then going to respiratory bronchioles in here. We'll get a better look at those, but we're going to terminal bronchioles to respiratory bronchioles. The alveolar ducts, the alveolar sacs. Alveolar sacs are more square-like, and the alveolar ducts are elongated. Each structure here, like here, and here, and here, and here, that's an alveolus. We're just seeing it in 2 dimensions. You have to imagine that it's in 3 dimensions. So this is an alveolus, this is an alveolus, this is an alveolus. So all of that. And so we'll look at that, and we'll look at it at much higher magnification. This is a bronchus. Again, if we see cartilage in the wall, it's a bronchus. We see a number of blood vessels here. That's a nice vein there, for instance. Here's our cartilage and smooth muscle and respiratory epithelium and bronchus. This is just a regular bronchiole. We could just call it a bronchiole. It's an airway, but it doesn't have any cartilage. But look at the nice smooth muscle. Again, sympathetic would cause these to dilate, let more air in. Parasympathetic causes them to constrict. We see the respiratory epithelium, we see the cilia quite clearly elucidated in a number of places there. And this is your respiratory epithelium. This is a electron micrograph, and we see that we have ciliated cells for here. We also have some non-ciliated cells in the bronchioles called clera or club cells. And this is an example. This is under an electron microscope image. And so here's the nucleus and some of the vacuoles and things in here, granules that can be excreted. These club cells can produce glycosaminoglycans that can help keep the airway open, kind of a lubrication almost. We need to say a word about these cells and why we're giving them 2 different names. These cells were discovered by a notorious medical doctor named Dr. Clara during World War II, and he discovered them by doing human experimentation on prisoners in the concentration camps. So it's just to illustrate that there's some research and things that we found out was done by very bad methodologies. And so they changed the name because they didn't want to honor this guy for that. So they changed him to club cells. But I sort of retained the name to at least tell you about them. We'll call them club cells, but it's important to remember, I think, that some things in science are done unethically, in fact, very horrifically. And this was one of those things that was done. So that's how they were discovered, but we tend to call them club cells now, although you'll see in a lot of sources they'll still be called Clara cells, because just historically that's what they've been called. So the ciliated cells are non-Clara cells. Those are just those regular epithelial cells. And then the Clara cells will produce basically lubricating glycosaminous glycans they produce primarily. This is a nice bronchiole, and you can see the smooth muscle really nicely elucidated. You can see the epithelium and some ciliated areas there, and the lung tissue surrounding it is pretty nicely seen there. This is a respiratory bronchiole. What's different? We have the lining, but look, we have some breaks in the lining. When we see these breaks in the lining, we say, now we're getting gas exchange. So breaks in the epithelium, now we say, that's a respiratory bronchiole. If we go back, notice here, there are no breaks in the epithelium. That's just a bronchiole, and in this case it's a terminal, being right before a respiratory bronchiole. But then we've seen some of these breaks, and we would call that a respiratory. This shows it a lot better. We're coming down the longitude. This would be a terminal bronchiole coming down. You see the epithelium lining it, and then right about here, do you see how it's breaking up the epithelium, like here and here and here and here? This, from that arrow, is called a respiratory bronchiole, because we do get gas exchange there. And so this is a respiratory bronchiole. Eventually it would go to an alveolar duct, and then an alveolar sac, and then an alveolus. So we see the transition happening there. Over here, of course, we see the alveoli, and we see alveolar sacs, like this little groupings of alveoli. There's alveolar sacs. If it's more elongated, like here, we call that an alveolar duct, for instance. And so alveolar ducts kind of transition into alveolar sacs, and then to alveoli. This shows this with a casting system, and then a scanning electron micrograph showing the bronchioles. And this shows a bronchial artery and vein here. There it is in color. This is an airway, and you notice the epithelium. Here's a vein and an artery. It's important to note that the pulmonary circulation is a low pressure circulation, so our arterial walls are not as thick as in the systemic arteries. So that's something to notice. We really have two sources of blood going to the lungs. The pulmonary circuit is carrying blood from the heart to get oxygenated, and it comes back through the pulmonary veins. Then we have the bronchial arteries and veins. That carries oxygen to the lung tissue itself. So it's supplying nutrients and oxygen to the lung tissue, not just getting reoxygenated. We are not, I've never found it that useful to try to differentiate between pulmonary arteries and bronchial arteries, or pulmonary veins and bronchial veins. We do label some differences, but we wouldn't expect you to know that they look very similar. There's really, I don't see much advantage in trying to differentiate them. But we do want to differentiate between airways, the bronchioles, and the arteries and the veins in general. again, notice that the artery doesn't have as thick of a wall as we might expect in the systemic system. Look at the lumen compared to the wall. It would be tempting to call this a vein, for instance, but this is an artery. We can see a bronchiole here going to a respiratory bronchiole and then an alveolar duct coming down the alveolar sacs. Here's a bronchus here. Again, here it is. This is a terminal bronchiole, and then it goes into a respiratory bronchiole. These are veins here. Very thin wall, but you can see there's a vein and there's a vein. And you can see there's labeled the veins and the respiratory bronchiole and all of that. This is an artery, again, it's not that thick of a wall, so it can be a little bit confusing. This shows you some clera or club cells, as we might more properly call them now. They tend to have this dome shape. Here we'll see them a little bit better. You see this dome shape, and the cytoplasm tends to be a little less intensely stained because it's kind of a mucousy secretion that they secrete, and so it doesn't pick up the stains so well. So these are club cells, we'll call them.
Are they a type of epithelium?
They're mixed in with the epithelium. So this is still respiratory epithelium, and a number of these cells have cilia, like here. The club cells don't have cilia, but they're mixed in with the ciliated cells. So all these cells have cilia, you see there. But once in a while, we'll see some like dome-shaped cells that don't have cilia. Look at this one here, probably. Squeezing there, a little bit of dome shape, and then the cytoplasm is a paler staining. So this is a bronchiole, and you see that same image in black and white. And again, a terminal bronchiole going to a respiratory bronchiole. This one happens to show a type of cancer called coat cell carcinoma or small cell carcinoma, this thickening here. I'll show it here and here. That thickening is abnormal. Yeah, so we'll look at the end, probably not today, but at the different types of cancer. We'll go over these in labs, so I'm just going to go over it fairly quickly. They're labeled. This shows it with the lung tissue, and again, we're going down to the bronchioles, the terminal bronchioles, respiratory bronchioles, alveolar ducts, alveolar sacs, and you can see them labeled here. Again, showing similar structures there, and we're going to be doing that in lab eventually. Shown here with the alveoli. The most common type of cells are these more flattened cells. Those are pneumocyte 1 or alveolar type 1 cells. Then we have the more rounded ones, which are alveolar type 2. They produce surfactant, and then we have some macrophages or dust cells as well. This is the blood showing the capillaries that comes around. This is a cast of the capillaries. There it is. Look at the capillaries. They wrap around the alveoli. Get a huge surface area, about 75 square meters of surface area. This shows you the larger alveolar type 2 cells, or pneumocytes type 2. They secrete surfactant. The more flattened, more common cells are type 1 alveolar or pneumocyte cells, and they make the structure. This is a little capillary in here. Capillaries come in through the walls, and then we have these flattened type 1 cells, and then the more rounded type 2 cells. These are macrophages, dust cells, and here we see some macrophages. These are the flattened ones, then more rounded, so type 1, type 2, type 1. And again, we'll be practicing that. These are capillaries, that's what the CA is. Here's a capillary with a big red blood cell and a platelet inside of it. Coming close there. This is a type 1 cell. Macrophages and type 1 and type 2 is shown over here. And we'll kind of look at those. This is pneumonia, where you've got a lot of fluid infested in there. And then we'll look at these clinical considerations. So we're out of time for today, but when we do last, we'll kind of look at these last few.